The overall goal of this procedure is to create a modular protein array that does not require a purification step, making it compatible with any protein library. This is accomplished by first generating synthetic genes through assembly PCR. Next, the synthetic genes are arrayed on epoxy slides. To fabricate the microfluidic device, use PDMS with silicone control and flow molds. Then the microfluidic device is aligned to the spotted DNA array. Finally, rabbit reticulocyte lysate is flowed into the microfluidic device and proteins are expressed. Ultimately, results can be obtained that show expression of thousands of proteins through fluorescent antibody labeling.
There are several advantages for using the microfluidic-based technique over existing methods such as protein microarrays. We microarray DNA and then use cell lysates to make proteins on the device. Thus, we do not require protein purification, and the proteins never dry. They remain fresh throughout the experiment. The microfluidic technique also provides higher sensitivity.
To fabricate the microfluidic device, expose the silicone control and flow molds to chlorotrimethylsilane vapor for 10 minutes to promote elastomer release after the baking steps. Prepare a mixture of silicone-based elastomer and curing agent in two different ratios of 5:1 and 20:1 for the control and flow molds, respectively. Pour the 5:1 PDMS onto the control layer. Degas the control layer and bake it.